Great, thank you very much. Thank you very much for inviting me. Digital twins is something that has its 15 minutes now. As Andy Warhol said, we will all be famous for 15 minutes. So if you work in this field, this is your time.
But 10 minutes for questions afterwards.
Fair enough. So what I will do is, I think a good part of my talk will be my version of what I think digital twins are. It's still a concept that a lot of people have questions about, have confusion about what's the difference between a digital twin and a model, things like that. And then I will tell you a little bit about some of the work we've done toward developing appropriate technology. Just by way of giving you a few sentences of background, so I come from pure mathematics, so I cover all bases at IPAM, having started in algebraic K-theory, module theory, topology. And for the last 25 years, I've worked in mathematical systems biology, I would say. And for the last 5 years, I've been at the Department of Medicine at the University of Florida, surrounded by experimentalists and clinicians. Let me first thank my colleagues who have done a lot of the work that I will tell you about. Just to give you an idea of the breadth of things we do, the gentleman on the left in the front row, his PhD is in algebraic number theory, and he is now an NIH-supported experimental program on immunology using mouse experiments. While I personally don't work at this time in cancer-related problems, at the upper row on the right, as you see Helen Moore. Many of you might know Helen Moore. She's worked on cancer in industry for a long time, and she's joined us for the last 4 years. So we do a breadth of different things, and of course, people might say then, that's a recipe for doing everything poorly. But anyway, that's what it is. We have collaborators. The work that I will tell you about, Lukas Böttcher from Frankfurt, he's working with us. Our immunology collaborator is Borna Merat. His lab is essentially across the hall from mine. We work with Kitware, a data science company. And a lot of the work we've been doing is with the working group on building immune digital twins of the Research Data Alliance. So it's a global effort in developing standards and so on. And a lot of this work has been possible through funding from various agencies. So what are digital twins? As I said, for a long time, there's been some confusion about what they are, but that confusion was settled to a large extent by a report that the National Academies of Sciences, Engineering, and Medicine commissioned in 2023 called Foundational Research Gaps and Future Directions for Digital Twins. They in particular gave a definition, which you see in this figure here. Probably every article on digital twins has this figure in it, and so you've probably seen it somewhere. And it says, well, a digital twin is two things. Twins are two things. You have the physical counterpart applied to medicine, that's a patient. You have some virtual representation, that's a computational model of some sort. And you have a bidirectional linking between the two. And once you have all of those, then you have a digital twin. So this virtual representation, one of the things that distinguishes medicine from engineering, for example, is that you have many different types of computational models that are suitable for different types of medical questions you're interested in. And that produces extra challenges. And then there's a whole other issue with data collection and so on. So this report in particular has a whole bunch of mathematical and computational issues in it. It's not domain specific, it's general questions. So if you're interested in the theoretical side of this, that report is, I would highly recommend it. So just to put it, just to put it in a medical context, you have the patient, you have a virtual representation of that patient, and you have, going in the one direction, you take data off of the patient over time, as you see in this graph in the middle, that essentially makes the digital copy of this patient evolve with the patient over time. And going in the other direction, you can use the digital twin to get information about, for example, prognosis or diagnosis or designing different interventions. So that's the setting. Let me just give you a few examples. The two examples I really like they're examples that are actually on the market. The one is the artificial pancreas, and that is the digital twin in this case is an ODE model of glucose metabolism, human glucose metabolism. It is calibrated every 90 seconds or so by measurements from a sensor, a subcutaneous sensor that takes glucose measurements. And the ODE model is coupled to a closed-loop controller that calculates how much insulin that person needs at the time and drives an insulin pump that injects insulin. And while it is not quite automatic, the patient still needs to input some information about what they're about to eat, whether they're about to exercise. And it does not work perfectly. It works pretty well. So every time, almost every time in a talk when I bring up this example, somebody afterwards will come up and show me that thing strapped to their waist, saying, you know, it doesn't work perfectly, but it really changed my life. And the other one I want to mention is a digital twin produced by HeartFlow. It's a company that does the following. You take CT scans of the vasculature in the heart that you use to simulate blood flow, and you can do things like risk assessment for aneurysms, for example, for a patient. You can do things like advice to a heart surgeon who is about to attach a graft for a blocked artery in terms of how you determine the optimal angle to minimize turbulence, things of that sort. It's sort of a digital twin. What it misses is, generally in applications, is it does not get updated periodically. It's usually a one-time thing, but you could certainly do it periodically. So let me give you just a couple of examples that really relate to cancer. There is one project that's described in this article here by the late Ilya Shmulevich. He has started, or he had worked for quite some time on a digital twin project in acute myeloid leukemia. And he was at a point where he was ready to essentially build a company to make that digital twin a commercial product. And a little over a year ago, he tragically died from that very same disease. And if you want to, so here's in this article, you find the description by Ilya. The project is continuing at the Institute for Systems Biology. So there's one. The other one I want to mention is Tom Yankelov. He has a digital twin, a working digital twin that they've been using in clinical trials that is used to decide how well therapy works, chemotherapy, other kinds of therapy. In, I think at the moment they do breast and brain cancer. And that is based on calibrating a pair of an ordinary and a partial differential equation from imaging data and making predictions. And that's described in this article here. This pair of articles comes from a workshop that we did a couple of years ago on problems related to digital twins. Here's a QR code for the web page of that. One of the things you'll find on that web page is YouTube videos of all the presentations, including Tomyankovs, and Ilya Shmuleviches. So it might be useful to check that out if you're interested. Now, just to put digital twin projects in context, if I take this loop in this National Academies report, which is often referred to as the NASAM loop, if I take it and I cut it and I string it out as a workflow, then what you see is basically this part here is what we all know how to do, is you build and validate the computational model based on some biology that may either exist or you may have to develop biology to get to a validated generic model of a particular condition that is of interest to the application for this particular twin. So it could be glucose metabolism in humans. And then comes the next step, which is model personalization. So then you calibrate the model to one individual patient, or if that is not possible, to at least a small population of patients. Yes, sir.
So I think kind of we are familiar all with this kind of model validation step, but we don't use the same kind of model, right? So if there's one person who's a PDE person has a certain model that is also validated for purpose, and another person is doing whatever stochastic processes, then I could have two digital twins that describe the same reality, and you would still call them digital twins, right?
Sure. Sure. If they serve the purpose you want, if they faithfully reflect what happens with a patient, then whether you do this with a Gaussian process or an ODE system is irrelevant. Yeah. And that's it's a legitimate question. Which model do I pick? And it could be determined by a number of different things. Which model captures best what's going on? If I have some spatial features that are important, as we heard about, then maybe I want an agent-based model or I want a PDE. If spatial features are not important, I can do something else. So then you have this model personalization, and then you have the application. You want to treat the particular patient. So if you look at it this way, it is what one typically would refer to as a bench-to-bedside project, right? And that's so that's great because you have this whole pipeline in one place. The challenge is, of course, number one, that now you need modelers, you need experimentalists, and you need clinicians, ideally to work together as a team. And probably all of us know how challenging it already is to have biologists and modelers work together. Never mind throwing in clinicians, because these three different cultures are extraordinarily different. And then I think, in the end, the biggest problem we face in making this into a sort of a scalable approach to personalized medicine is a cultural problem. The other challenge is, of course, if you now try to get a project like this funded, you will have tremendous difficulties because, for example, if you go to the National Institutes of Health, there is no ready-made program that you could apply something like this to. Even larger projects, I don't know if you're familiar with like PO1s, which are sort of three related projects put together. These projects sort of need to be able to stand on their own. But once you take this apart into three pieces, you don't have a rationale for each one of these pieces. So we're trying to contort ourselves into pretzels right now to write one of those PO1s for a digital twin for sarcoidosis. So as the title says, we focused on agent-based models. We get started in modeling the immune response to fungal infections in the lung, which are spatially very heterogeneous. Space makes a difference, just as it makes a difference in cancer, as we heard from Dominique earlier. So let me just point out some examples I really like. This is from the lab of Denise Kirschner for the last 25 years. She's been working on modeling granulomas, the development and treatment of granulomas in tuberculosis. So this is at the tissue level, it is an agent-based model. At the intracellular level, it is a differential equations model. So it is what Dominic earlier called a stochastic hybrid model, multi-scale model. So it has a lot of the complicating features, and I know Denise is now refashioning her model to also work on cancer-related applications. So one of the things we're working on is a digital twin for pneumonia patients, essentially a decision support tool for critical care doctors in the intensive care unit to decide how to best treat pneumonia patients. So this we started really by trying to find a host-centric treatment against fungal infections. The standard treatment is antifungal medication similar to antibiotics, and the problems are the same. Antifungals don't work increasingly, just like antibiotics don't work. And the reasons to some extent are the same. Antifungals are used prolifically on various agriculture products, and bugs develop resistance. And but this turned into sort of a digital twin type project. And I just want to make one comment, since I came up a couple of times, small models versus big models. And I'm all for small models, but this is a big model, relatively speaking. This has maybe 80 parameters. And if we had a specific question that we wanted to answer, we would be able to build a much smaller model. Now, what we did with this is we applied it to a number of different things. So, for example, we were interested in looking at the phenomenon that immunocompetent patients, people with an intact immune system that had COVID infection, developed also invasive aspergillosis, which only happens in immunocompromised patients. And we had other applications. And for each one of these applications, sensitivity analysis shows there's half a dozen parameters that are really relevant and the other ones are not. Except for each of these applications, that half dozen parameters is different. And I think that's a good argument for building more complex models when it's appropriate, when you don't need a single purpose model, if you like. Okay, now this of course is Paul Macklin's Physicell, which I'm sure a lot of you have seen. So Paul has basically set up a platform that incorporates all the biophysics that you would need for setting up a tumor, and then you can customize it to particular applications. And we're talking with Paul and his team now about incorporating some of the tools that I will mention later on that can make this into a model that goes into the digital twin framework. So let me first talk about one of the directions of the digital twin, and that is from the digital twin to the physical twin. So that's control, and Eduardo, I included this slide specifically for you. Somebody pointed this out to me, and it makes sense to me, but I know nothing about control, and you know everything about control. So hopefully, maybe at the end we could get your opinion. So basically, what Ivan Yenko says, and he's sort of one of the fathers of deep learning theory. What he says is essentially, if you want to control complex systems, then modern control theory based on differential equations is just not going to do the trick. And you know, of course, humans are decidedly complex systems.
Do you know what he means by paths? He means solutions of the equation? Is that what the word path means?
I would guess. I really don't know.
Okay.
So if not differential equations, then what? That's a negative statement. What's the positive statement?
Yeah, so I have to admit I did not read the article.
Just put it there to
And I just said, you know, I'm just going to let Eduardo deal with that. So, okay. Okay. All right. So what we would like to do is we would like to use an agent-based model for control purposes. Now, if you know about agent-based models, they are great. They are very flexible. They can allow, for spatial heterogeneities. They are easy to understand. Non-modelers can build them, but they have huge drawbacks. One is that there's no standards. If you go into the literature and you look for a paper with an agent-based model, then 99 out of 100 times you will not be able to take any kind of code from anywhere and run it and be reasonably sure that what the code does is actually compute what the authors describe as being the model. And this is particularly a problem if you have stochastic models. So we used one model in the things I'm going to show you in a minute. It's a very simple predator-prey model of wolf-grass sheep. So it's wolf and grass and as a resource and sheep. It's implemented in NetLogo. It's very nice, very simple. It's a stochastic model. And so for our purposes, we re-implemented it. And so we have 4 different implementations of this model. And to the naked eye, so to speak, they look identical. Now, my colleague TJ Sego and his collaborators, they have worked on reproducibility of stochastic models. And they've developed rigorous tools to measure whether 2 stochastic models are the same. And based on his tools, those 4 models are different. They have different dynamics. So that's an additional complexity. But probably the biggest complexity, if you want to use agent-based models in medicine, is that what you need as an initialization for example, one of Paul Macklin's tumor model, where you have, let's say, 10,000 cells, is that you need an initialization that has information about every one of these 10,000 cells. And of course, you will never be able to measure that, even remotely. And that's one of the challenges that we face, is that you can't measure all the stuff that you need. So last year we published 2 papers that address this issue. The first one looks at control via surrogate models, and we looked at a range of different surrogate models to use. And I'll tell you in a minute what they are. And we applied those to 2 examples. One is this wolf-grass sheep model that I told you about. And the other one is an agent-based model that's an instantiation of a biochemical reaction network, a metabolic network. And it was designed specifically to foil some of the surrogate models that we built. And so the idea is, of course, you take a surrogate model for which you know how to do control, say a system of differential equations. There are standard optimal control methods. They're all in Eduardo's book. And you can then find optimal controls, ideally lift them to the agent-based model, and go from there. So in the other paper, we looked at control by using artificial neural networks that are directly applied to the agent-based model. So let me show you what the outcome was. First, we looked at different surrogate models. So we looked at mechanistic approximations. So these are ODEs that are sort of built by hand for a specific control objective. We had general mass action models as surrogate models. We had S systems, so very general approach, quadratic Taylor approximations. And the control problem that we tried to solve with all of these models was, in this wolf-grass sheep situation, one of the things that we did is steady state control. You want to move the model from one steady state to another one by, say, changing the mean number of wolves or sheep from what it is to something else. And so a control input was at each update, you reduce wolves and sheep by a certain number, right? And so the goal is to get to the new steady state with a minimum number of animals that you remove. Okay, and here's what you see what happened is, my guess would have been that the mechanistic model would obviously do the best. But it actually does fairly poorly. And the S-system approximation, a very phenomenological generic one, does the best in this case.
So the goal is where it says ABM? Where's the goal?
So if you do this by using the ABM directly, and in this case the ABM is easy, so you can optimize directly, then you get in the parameter space what the number of sheep and wolves you remove at each time step is captured here. And so this is the optimal solution. And here you see how close these other ones got to it.
So what actually is the mechanistic model corresponding to net logo simulation?
So you build a small system of differential equations that capture, this is really a Lotka-Volterra.
There's 3 equations?
I'm sorry?
2 equations, 3 equations? The grass changes? Or?
The grass changes. So it's
3 equations.
It's 3 equations. So it's a Lotka-Volterra model with 3 equations.
But it somehow corresponds then to a mean field model without space, right?
Yeah.
So the mechanistic is automatically kind of a non-spatial model. And does this hold for many of these kind of non-central models?
No, this particular one is a spatially homogeneous model. So it's...
Oh, no space even in the ABM?
There's space, but there's no spatial heterogeneity, really. It's a uniform.
They eat only their neighbors, or do they not?
They eat only their neighbors.
Then you can't do it with the mechanistic
and they move
mean
around.
field. It's not surprising that the mechanistic non-spatial model doesn't do well, right? Because it cannot capture the nearest neighbor interactions.
So each one of them moves. So it moves at a... Oh, I'm sorry, no, they don't just eat their neighbor. At each step they move with a certain distance and in a certain direction. So it's not a nearest neighbor setup.
But they can only eat their nearest neighbor. Right?
They can only eat, they can move potentially quite far. Yeah.
The wolves and the sheep eating the sheep? Is that what it is? The bird eating the
I wouldn't expect an ODE to capture a spatial ABM.
Do they move uniformly? Like with equal...
They move uniformly, yes.
With equal probability everywhere?
Correct.
But then it should capture it, maybe, right? Because then it's like not really...
Or moving like to a randomly chosen grid
then
point,
it's-
or just to the neighboring grid points?
No, they can move randomly.
So then it's...
Okay.
You just
And then the- okay.
No, it's
Yeah.
fine,
All right.
Anyway, so in the second paper, we looked at
Sorry, still in the first paper then. Sorry, I'm lost also about what the model is. So what is the ABM model in that case exactly?
so the agents are wolves, grass, and sheep.
Right, and there's a number in
Grasses.
each place in a grid
Correct.
with periodic boundary conditions or something?
Correct.
Okay, and then they can move to some other, this discrete time now, right? And they can move to the presumably neighboring thing with some probability, and then the interactions occur only at that pixel,
Correct.
only at that cell.
Yes.
Okay.
Yeah. Okay. So the next in the second paper, we looked at an artificial neural network controller. So it takes as input population counts and gives as output essentially numbers of animals to be killed, if you put it that way. And so this was trained on a number of trajectories with and without control, just like in the previous paper. And as you can see, it does extraordinarily well. It really hits the optimum on the
So how
node.
did it get rewarded? Sorry, I don't understand.
How do you
So
get-
it's an open loop network, right? Well, it's not open loop, sorry. It's measuring the animals, they put it in closed loop, and then you're simulating this and you have a loss function
loss function, correct?
that measures distance to the optimum or something.
Correct.
I see. Okay.
Yep. Okay, so in this simple case, the neural network controller does extraordinarily well. And it does extraordinarily well in some other cases, but in some yet other cases it fails. For example, in this metabolic network that we used, it fails fairly seriously.
So is that because you didn't have enough neurons, or what, I mean, how big was the network?
No, I mean, for example, one of the things that it did not capture well at all is any sort of saturation.
What activations were you using? ReLUs?
I don't remember. We in the same paper, what we also tested was neural ODs, as we just talked about at lunch. And so neural ODs, those are differential equations with a neural network as the right-hand side. And there too, with this is on this metabolic network. This is a picture of the loss function. These blue dots, that's the ABM, and you can see that the loss function for the neural OD does better than the other one. So at least for these models, the neural network-based surrogate models do quite well. The problem is that there's sort of very little that can. I mean, if you would like to make a general statement about some class of agent-based models for which this works or that works, it's difficult. And if you want to test, say, a bunch of published models, you essentially have to re-implement every one of these published models. So it is a major production to do that. And we, over time, we sort of are collecting a suite of test models implemented in Python that are more easily used. So I'm almost out of time. So let me just go to the other direction, and that is how you personalize a model to a particular patient. And there, the main problem, or one big problem, is precisely this. The fact that you need a whole bunch of information to initialize an ABM. We'll call that, you need a microstate with a whole bunch of detailed information. But in most cases, all you can measure is a macrostate. You can maybe measure the total number of immune cells of a certain type in the blood or in fluid you get out of a lung, or number of cells in a tumor. And so that's a problem that we have addressed. So a microstate would be a full initialization of the ABM. The corresponding macrostate would be consisting of model parameters, model states that are parts of the model that are not represented in great detail. For example, your model might have a liver compartment that simply delivers a particular enzyme, and all you get from the liver is the concentration of this enzyme, and then summaries of microstates. So the problem that you face is essentially an ill-posed inverse problem. You have repeated partial measurements. These are the data you get off of a patient. For example, a patient with certain cancer, you come in and you make measurements every 3 months, 5 months, 6 months, and you want to, from that, you want to calibrate this ABM and simulate for the purpose of forecasting. Just to say the approach without going into any details, we've used at the macrostate level a common filter type approach. So common filters, if you had a system of ODEs, that would be a good approach for, in this setting, it is not so straightforward to use. Common filters have certain assumptions. You need a state space model. Agent-based models are not state space models. State space changes, you have birth-death processes and so on. It needs to be linear. You need to have certain distribution requirements and so on. So it's two things. Basically we map the microstates up into macrostates and use a common filter type process up here. And once we have a macrostate, you need to synthesize a microstate that is a likely state that gives you that macrostate. And that is reasonably straightforward for spatially fairly homogeneous models. If you have spatially heterogeneous models, it becomes quite complicated. And we have solutions that are, I would say, ad hoc solutions that work in some cases, but definitely don't work in other cases. So so there is just in this approach, there's a lot of work to be done, both for control and personalization, or data assimilation is a more technically appropriate term. We've been working with a number of AI, generative AI tools, variational autoencoders, and so on. I'm generally very skeptical about AI type things, and it is not my first thing to go to, but in the work that we've done, I've become convinced that in certain situations they work extremely well. One needs to be careful if you were to use an AI-based model as the underlying computational model for a digital twin, because whatever data you pull off of your patient is going to be very, very little. And so that is an inherent limitation. So that's what I would say is one open problem. Uncertainty quantification in all of these things is another problem that certainly in the setting of agent-based models we have not begun to think about. One of the things one might consider is the analog of large language models, where instead of language-based rules you have physics, chemistry, biology-based rules. I think that could be quite promising. Finally, in order to really make this approach scalable, you need to develop a general platform that people can come and sort of plug in the particular application they have, rather than having to buy a kit, so to speak, and then put it all together. So that's another problem, I think, that has a lot of mathematical issues attached to it. And finally, I would say the biggest problem is that ultimately a digital twin is a device. It is a device that needs to be used. So whatever your digital twin does, it has to run on a doctor's tablet or some similar kind of device, and it has to be quick and it has to be useful.
Why does it have to be edge? Why can't it be cloud-based?
I mean, it can be wherever, but the doctor needs to access it.
Right, okay, but just the GUI. But...
Yeah, no, I'm not saying it should run on the
Okay. You use the word run.
Yeah, I'm sorry.
Okay.
No. It needs to be accessible.
Okay.
I mean, if you ask a doctor, where would you like to have this digital twin? They will say, well, it needs to be in the patient's chart. It needs to be in the EPIC chart. Right? So that by itself is a humongous non-mathematical problem. If you get to change anything in EPIC. So, okay, I'm done. I ran way over. I apologize.